Good morning. All of you have joined. Raibag class, settle down quickly. Settle. Class 2 Raibag. Kelly Raibag class 2, settle quickly. Okay, who is this? Which class you are telling about? Oh, late aita. This was your break, kya? Okay, Nipani class 19 and 20 settled? All set? Ha, na, say something. Do actions at least so that I can see you, understand you. Bandaralwa, class 19 Nippani is settled. Okay, class 20 is also set. Okay, fine. Replication, we are going to start replication. Meanwhile, start reading. Other classes are settling down. Joseph, you also start reading replication. Raibach, New PU College, please settle. Okay, fine. Let's not waste much time now. Oh my god, what is this? Anybody? What is this? Lighthouse? Sure. How many of you been to Eiffel Tower? ಎಷ್ಟು ಜನ ಹೋಗಿದ್ದೀರಾ? At least in dreams, in phone. Must have been, na? Eiffel Tower ge. Okay, so I am going to start with our next concept, that is replication. Okay, the very first proof of replication, that DNA replicates in a semi-conservative manner. Who gave it? Who gave the proof that DNA replicates in? Yes, who was it? Messelson and Stahl. The Messelson and Stahl were the scientists who discovered, but along with that, there were other approaches also. Check if you find out the name scientist Taylor, who worked on Vossia fabia, a bean plant. It must be there in your book also. He has Taylor experiment on the fabia is a bean plant where they found out the using thymidine. Irbek nodi, check in your NCRT last paragraph somewhere. One first Messelson and Stahl experiment ಆದ್ಮೇಲೆ ಎಲ್ಲಾದರೂ ಇದೆ. Check. Taylor experiment on VCF ಅಂತ. It should be there. If it is not there, then I'll tell you. ಇದ್ರೆ ಒಂದು ಸ್ವಲ್ಪ ಅದನ್ನ ಅಂಡರ್ಲೈನ್ ಮಾಡ್ಕೊಳ್ಳಿ. Please open your NCRT textbook. Messelson and Stahl ಆದ್ಮೇಲೆ VCF fabia ಮೇಲೆ ಒಂದು experiment, Taylor experiment ಆಗಿದೆ. 2 or 3 sentences must be there. Joseph, can you check? ಸಿಗತ್ತೆ. You'll find it out. Okay, at the first place now, let me start to understand you that replication is simply copying of DNA, right? We know that. Simple replication is nothing but a copying of DNA. Now my question is, when do you think the DNA has to undergo the copying? Why the DNA needs the, or what is the necessity of a DNA to be copied? Anyone? DNA copy ಯಾಕೆ ಆಗಬೇಕು? Why this replication is needed? Copying of DNA. Why? Why and when? Come on, basics. Nobody knows this. Okay, when does the replication happen? Try. Replication ಯಾವಾಗ ಆಗತ್ತೆ? I'm not able to hear if somebody's asked. ಯಾರೋ ಒಬ್ರು ಮಾತಾಡ್ತಾರೆ ಅಂತ ಗೊತ್ತಾಗತ್ತೆ, but can't hear the words clearly. Correct. That means during the S phase of cell division. So this answers your, during the S phase of cell division, replication happens. Why? Nothing. very, very far voice. Raibag, new PU Raibag, anyone? Why? Why you think the replication is done? Nipani? Anyone from Nipani College? Simple logic, people. When one cell has to divide to give you two cells, the DNA from this has to make a copy of it so that it can give copy equally to the daughter cells. Correct? Cell division, that is the reason, no? That in cell division only it becomes the copy. So you should know this basic. Cell will not do replication continuously. For example, my skin cell, whatever cell is present in the skin right now, every DNA from each cell is not going under replication. It is simply doing transcription and translation protein synthesis. Replication ಯಾವಾಗ ಮಾಡಕ್ಕೆ ಸ್ಟಾರ್ಟ್ ಮಾಡುತ್ತೆ? ಸಪೋಸ್ ಏನೋ ಒಂದು ಕಟ್ ಆಯಿತು, ಏನೋ ಒಂದು ಗಾಯ ಆಗಿದೆ, when you want to heal it, when you want to patch it, the cells from there will start to divide. Now when you have to divide, you need a copy of DNA, then the replication starts. And when the division is stopped, replication is also going to be stopped. Understood this basic thing? Next, now, what is the machinery? What are the different things which is needed for replication? We saw what is replication, when is replication done, why is replication done, correct? Now, the length or the time period to copying the DNA varies from organism to organism. For example, E. coli, they have given E. coli replicates within 18 minutes itself. And the speed of the enzyme is where it can put almost 2000 base pair per second. Can you imagine 2000 base pair per second? You can't even blink an eye in a second. So imagine it is putting the enzyme. Which enzyme is required for replication? People start answering, I will understand that you have not understood this chapter at all. Idune artha agate amale. What are the requirements? First enzyme, what is needed for replication? ನಿನ್ನೆ ಹೇಳಿದೆ ನಾನು ಓದ್ಕೊಂಡು ಬರ್ರಿ ಅಂತ DNA dependent DNA polymerase. Then you need ORI. Then you need DNA ligase. Actually, there is one more enzyme which is not there, but I have to tell you because you will opt for NEAT also, helicase. Then you need what? You need DNTPs. What is DNTP? Anyone? What could be DNTP? You know this. ನಾನು ಈಸಿ ಲ್ಯಾಂಗ್ವೇಜಲ್ಲಿ ಹೇಳಿಬಿಟ್ರೆ ಓ ಮ್ಯಾಮ್ ನಮಗೆ ಗೊತ್ತು ಅಂತ ಹೇಳ್ತೀರಾ. You know what is DNTP. Try to split. For example, ATP is a DNTP. GTP is a DNTP. TTP and UTP. What is this? GTP is also a DNTP. Logic? Use your head. No? There is the word in your book called as deoxyribonucleotide phosphate. DNTP. Deoxynucleotide phosphate. Deoxynucleotide triphosphate is nothing but DNTP, which is nothing but your adenine, guanine, thymine, and cytosine. Purines and pyrimidines. So for replication, you will need nucleotide also, right? So deoxynucleotide triphosphate, DNTP, deoxynucleotide triphosphate. This word is there in your book. You must have not read that. It is Nimta Padna. Odbeku neevu. Okay. So what are the requirements needed for replication? You need helicase enzyme, DNA dependent DNA polymerase. Then you need ORI site. ORI site is a site of origin or the point at which replication initiates or starts. Then you need a ligase enzyme, and finally you also need the DNTPs, deoxynucleotide triphosphate. E word book ali ide, guarantee. Anyone search and tell me. And DNTPs are nothing but your nucleotides, adenine, guanine, thymine, and cytosine. Purines and pyrimidines ge idannanta commonly ಹೇಳ್ತೀವಿ ನಾವು. Okay, so now you can imagine how fast this DNA dependent DNA polymer is. Anybody can tell me why it is called DNA dependent DNA polymerase? Polymerase, the name polymerase means what? One enzyme, the one which goes on adding multiple polymers of nucleotide. So DNA polymerase again, full form is DNA dependent, meaning DNA strand ಅನ್ನ ಯೂಸ್ ಮಾಡಿಬಿಟ್ಟು DNA ಮಾಡ್ತೀವಿ. You are making the DNA from the DNA, so DNA polymerase. This is confusing. I know, I understand the chapter is confusing, chapter is huge. I am going to tell you it in a very detail. Please pay attention. This chapter is already done to you. Andru artha agila. Nobody is able to answer me. So at least pay attention when I am trying to tell you. DNA dependent DNA polymerase meaning using the DNA as a strand, the next polymerase makes the new DNA. If the polymerase was making RNA, then the enzyme names changes as DNA dependent RNA polymerase. For example, in transcription ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, DNA ಇಂದ mRNA ಆಗುತ್ತೆ, ಅವಾಗ RNA polymerase ಅಂತ ಹೇಳ್ತೀವಿ. ಈಗ DNA ಇಂದ ಇನ್ನೊಂದು DNA ಮಾಡ್ತಾ ಇದ್ದೇನೆ ನಾನು, so DNA dependent DNA polymerase. Using the DNA template, the new DNA copy is made by the enzyme called as polymerase. Is it clear? Don't sit as if you have come to watch a movie, people. I need an interaction. Illa naan helidmelanu artha agalla, you will not be able to attempt any question from this chapter then. The loss is yours, so speak up. Fine. Let us go to Michelson and Stahl experiment and see what did he try to prove. Okay. Can anybody try to explain how the semi-conservative was proved by him? Whatever you know. See, ನೀವು ಮಾತಾಡಿದ್ಮೇಲೆ ನನಗೆ ಗೊತ್ತಾಗತ್ತೆ how well you have understood anta. Then I can easily again repeat if I feel that you have not understood. So try. What did Michelson and Stahl experiment prove? What bacteria he used? They have used the E. coli only. So initially E. coli were allowed to grow on the medium which contained nitrogen. Yeah, isotope first of all. Lighter isotope. Right? N15. So first of all, E. coli were allowed to grow into the medium that contains N15 CSCl. So what will happen? Initially all the E. coli will have the DNA which has N15. So here you can see the DNA that has N15 in its N andre ಏನು nitrogen. DNA ಅಲ್ಲಿ nitrogen ಎಲ್ಲಿ ಬರುತ್ತೆ? Where this will go and insert? Good job. Base pair. Yes, base pairs are made up of nitrogen bases, meaning there is a nitrogen compound in the base pair. Al hogi a medium, in this medium there is a nitrogen present. So DNA copy ಮಾಡಬೇಕಾದ್ರೆ ಆ nitrogen ಅನ್ನ it will try to put or incorporate itself inside the segment of DNA. So at initial time, the all E. coli will have their DNA which has N15. Now, when you're allowing this DNA or the E. coli to grow for next generation, E. coli almost take 20 minutes, right? So after next 20 minutes, you will now change the medium where N14 would be incorporated. N14 and N15 are radioisotopes, simply the light and the heavier element of the nitrogen. So N15 inda iga ಏನು ಮಾಡಿದ್ರು, change the medium in which N14, nitrogen 14 is present, the lighter 14. Now what will happen? This nitrogen 14, N14, was seen to be incorporated in a way where one strand considered was 15 and one more strand which was seen was 14, which was a hybrid. andre all the E. coli cell after first 20 minutes will have one strand which has 15 nitrogen, the nitrogen containing N15, and another strand which has the nitrogen containing N14. Now to cross check this, illé prove ಆಗುತ್ತೆ actually semi conservative ಅಂದ್ರೆ ಏನು, one parental strand remains same and the daughter strand is formed against the parental strand. illé prove ಆಗುತ್ತೆ, but fine, it has to again cross checked. So for again next 20 minutes if it is kept and analyzed, aವಾಗ ಏನಾಯ್ತು, this parental strand had N15, N14, now the new strand has N15 and N14. See, I'll draw it if you are not able to understand. Here, let me start from N15 only, better. Suppose this N15 and N15 both strand are there. 20 minutes aadmele, because the medium is changed, this medium contains 14, the new strand here, I'll change the color, will be N14. Again new strand here would be N14. So you get hybrid, this is also hybrid, this is also hybrid. Now keep it for again next 20 minutes. Again, athwa now it will become 40 minutes, let's say. In your book it is 40 ala, lastly. And the medium is same, N14 a medium ide, medium is not changed. Iga next time ಏನಾಗುತ್ತೆ, how this will come here now? Now what will happen? See, this I am drawing here down. If ಎರಡು strand, this first strand N15, then against this first strand you will have the new medium has N14, so it will be like N14 only. Then when this act as a parental strand, so this will be N14 parental strand, new strand would be N14 itself. Then when this is acting as a parental strand, avagu N14 e barutte. And lastly, when this is acting as a parental strand, that time N15 and N14. So now you got more of what? Only this is hybrid, this is hybrid, these are pure now. These are pure of N14. So as again, matte inon 20 minutes itre, let's say 60 minutes it bitre, what will happen? Either baju, then you will have one more here, N14, 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 N14. So again, number of hybrids are going to get reduced and the number of these N14 get accumulated, suggesting that parental strand was consistently kept. A parental strand opposite only, you are going to get the new strand. And this, when it was done using the cesium chloride density gradient andre yeno, the lighter and the heavier, if you see here, this is a heavier isotope. This is heavier isotopes. So test tube alli adanu kedage hogi settle agutte, it will be settled at the bottom. This DNA will settle at the bottom. When there is a hybrid, andre one strand N14 ide, ಇನ್ನೊಂದು strand N15 ide, that band is seen little bit upper because light strand bantu, one heavy strand bantu. Hangagi the band would be seen little bit upper ways. And then adadmele, if you go on seeing other things, 50% illi uldide, parental strand, 50% new strand. Now 70% new strand and 25% parental strand. What does this mean? That parental strand went on reducing as the number of copy of DNA went on increasing. And second conclusion in Bharata, that appearance of this hybrid bands suggests that N14 and N15, both of them are present in a particular band, which means whenever there was a N14 incorporated, N15 was used as a template or a copy. Iga suppose ನೀವು ಜೆರಾಕ್ಸ್ ಮಾಡ್ಲಿಕ್ಕೆ ಒಂದು ಏನೋ ಪೇಪರ್ ಒಯ್ದಿರ್ತೀರಾ, ಕರೆಕ್ಟ್? ಆ ಪೇಪರನ್ನ ಫಸ್ಟ್ ಟೈಮ್ ವೆನ್ ಯು ಕೀಪ್ ಇಟ್ ಆ್ಯಂಡ್ ಮೇಕ್ ಅ ಜೆರಾಕ್ಸ್, First paper is called as parent. Now when you get a copy of it, that is daughter. Iga, what you can do when you make a next copy, ಎರಡು xerox machine ಅಲ್ಲಿ ಈ parent copy ನು ಇಡಬಹುದು, ಇದನ್ನು ಇಡಬಹುದು and make a copy of it. Similarly, same thing happens with the DNA also. So this was the proof how the DNA is going to replicate in a semi-conservative manner. Full conservative or semi-conservative, full conservative ಆಗಕ್ಕೆ ಸಾಧ್ಯ ಇಲ್ಲ. Full conservative means ಇಡೀ parent strand is retained ಅಂತ ಅರ್ಥ ಆಗುತ್ತೆ. ಹಾಗಾಗಲ್ಲ. Semi-conservative means whenever the new strand is formed, one strand is parent, one strand is daughter. So cells divide ಆಗುವಾಗ, whenever cell will divide here and give its DNA, one would be called as parent strand, one would be called as daughter strand. So ide cell, when it makes the two copies, one goes in one strand, another goes in another strand. Athara. Understanding? This is a semi-conservative model, what Messelson and Stahl explained. Nipani, Rybak, Joseph, all of you are clear?
Yes, ma'am. Yes, miss.
Okay, any doubts? Fine. If no doubts, then I'll start asking question, and when I ask question and if you don't answer, erade artha. Either you have not understood anything or you have understood everything. So if you have understood everything, please answer. Now, I told you that requirements, you need DNA polymerase, correct? Then I told you that you need helicase also. Now this is a new enzyme, it is not there in your book, but I want you to remember questions baratte, NEET level questions baratte illi. What is this is, suppose this is your DNA, okay? Iga DNA mele DNA polymerase will identify this site, ORI site, called as origin site, the point where replication starts or initiates. Correct. So let's say, or let us assume that this is the point or this is the sequence where replication has to start. But DNA is a coiled structure. DNA is a coiled structure. Now this coiling or this complementary strands has to get separated. Then only you can open up. Idana separate ಮಾಡಬೇಕು. Then only you can open up like this Y fork. Who will open this? Opening means what? Illi enittu, they have bases. Correct? ಈ bases ಮಧ್ಯದಲ್ಲಿ bonds ಇದೆ. These bonds has to be broken. Helicase enzyme is actually used to separate the strands. This separates the two complementary strands by breaking the hydrogen bonds. Understanding the function of helicase, it will start breaking these hydrogen bonds, and therefore you will get the open Y fork. This diagram is there in your book. Ee thara illa, it is little bit more open ಮಾಡಿ ತೋರಿಸಿದ್ದಾರೆ. They have opened it like this. They have opened it like this, simply to give you more idea. So iga imagine there is a rubber band, twisted rubber band. ಆ ರಬ್ಬರ್ ಬ್ಯಾಂಡಿಗೆ ಅಥವಾ ಲ್ಯಾಂಡ್ಲೈನ್ ಫೋನಿಗೆ ಹೆಂಗೆ ಕಾಯಿಲ್ಡ್ ವೈರ್ ಬರ್ತಿತ್ತಲ್ಲ, so that coiled wire or that coiled rubber band, when you're opening at one point, when you open at one point, ಅದರಿಂದ ಟೆನ್ಷನ್ ಕ್ರಿಯೇಟ್ ಆಗುತ್ತೆ at behind. Here you will get the stress or tension. ಆಗತ್ತಲ್ವಾ? Imagine, take a big rope, ಎರಡು ಒಂದು ದುಪಟ್ಟ ಅಥವಾ ತಗೊಳ್ಳಿ, ವೇಲ್ ತಗೊಳ್ಳಿ, ಅಥವಾ ಟೇಕ್ ಅ ರಬ್ಬರ್ ಬ್ಯಾಂಡ್, ಟೈಟ್ಲಿ ಕಾಯಿಲ್ ಮಾಡಿದೆ, ಇನ್ ಬಿಟ್ವೀನ್ ಯು ಓಪನ್ ಇಟ್ ಅಪ್, ಹಿಂದ್ಗಡೆ ಮತ್ತೆ ಮುಂದ್ಗಡೆ ಜಾಸ್ತಿ ಸುತ್ಕೊಳ್ಳುತ್ತೆ. ಕರೆಕ್ಟ್? ದಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಸೂಪರ್ ಕಾಯಿಲಿಂಗ್. This is called super coiling. Means this will become more tight, like this, it will become more tight because you are opened up here. Understanding? Can you imagine this? when there's rubber. I don't have anything to show you people. Hang torusli nanu. Can you see this wire, all of you? Okay. Wait, I'm trying to show you something. This is something like this DNA. ಈಗ ನಾನು ಏನು ಮಾಡಿದೆ, ಐ ಓಪನ್ ಇನ್ ಬಿಟ್ವೀನ್, ಇಲ್ಲಿ ಓಪನ್ ಮಾಡಿದ್ದೀನಿ ನಾನು. ಸೊ ಇಲ್ಲಿ ಜಾಸ್ತಿ ಸುತ್ಕೊಂತಿದ್ದು, ಇಟ್ ಬಿಕೇಮ್ ವೌಂಡೆಡ್ ಹಿಯರ್ ಟೂ ಮಚ್ ಆ್ಯಂಡ್ ಹಿಯರ್ ಆಲ್ಸೋ ಇನ್ ಡೌನ್ ಬಿಕಾಸ್ ಐ ಓಪನ್ಡ್ ಹಿಯರ್. ಸೊ ಇಲ್ಲಿ ಟೆನ್ಷನ್ ಬೀಳ್ತಾ ಇದೆ. ದರ್ ಇಸ್ ಅ ಪ್ರೆಷರ್ ವಿಚ್ ಇಸ್ ಫಾಲಿಂಗ್ ಹಿಯರ್ ಆ್ಯಂಡ್ ಹಿಯರ್ ಆಲ್ಸೋ ಬಿಕಾಸ್ ಐ ಹ್ಯಾವ್ ಓಪನ್ಡ್ ಇಟ್ ಹಿಯರ್. ಓರೈ ಸೈಟ್ ಇಲ್ಲಿದೆ. ಸಿ ಇಲ್ಲಿ ಜಾಸ್ತಿ ಸುತ್ಕೊಳ್ತಾ ಇದೆ. That is what I'm trying to explain you. The DNA will get more twisted, super coiled. This supercoiling is released by the enzyme called gyrase. Again, a new term. Idu yak ಹೇಳ್ತಾ ಇದೀನಿ, if anybody is opting for NEET, you need this information. Okay. Then, okay, so what we did at the ORI site, the DNA strands are opened now. Opened by the enzyme helicase. Helicase yen ಮಾಡ್ತು, it will start breaking all the bonds, bases, in between bases, whatever bonds are there, that bonds are being breaked here, and this becomes a Y fork. This is called as Y fork, or it is also called as replication fork. Y fork or replication fork. Then our DNA polymerase will come and sit here and start synthesizing the new strand. Now, DNA polymerase will synthesize the new strand in which direction? The direction of DNA polymerase? It's there in the book. Everybody is looking at me like this. What is the direction of DNA polymerase? 5', 3', and 3', 5'. ಹಂಗೆ ಹೇಳಲ್ಲ. ಸಿ ಯು ಶುಡ್ ನಾಟ್ ಟೆಲ್ ಲೈಕ್ ದ್ಯಾಟ್. ಓಕೆ ಫೈನ್. ಇಲ್ಲೇ ಹೇಳ. ಓಕೆ ವೇಟ್. Suppose the same strand, I'm taking it here. Okay, let's say this is your 5 prime to 3, no, ulta. This should be 3 prime to 5 prime, 3 prime to 5 prime. It hinge irate. 3 prime means this is your 3 prime and 5 prime. No, ulta. Fine. Now, polymerase has the direction to go into 5 prime to 3 prime direction, meaning it is going to synthesize like this. This is 5 prime to 3 prime direction. But the template direction is always 3 prime to 5 prime. What I'm trying to say is, when enzyme comes and sits on this both the strand, same diagram, okay? When enzyme comes and sits on this both the strand, this enzyme will move forward and go on adding the DNTPs here in the direction of 5 prime to 3 prime. Okay, so what happens when the enzyme has direction to go in one direction? The strand would be synthesized continuously without any gap. That is why we say that one strand becomes continuous, or it is also called as one more name, leading strand. Continuous strand or leading strand, you should say. My God, people, I really want you to respond, okay? Because ishtu detail naan ee chapter hodre, when am I supposed to cover your questions? Next class, at any cost, I want you to read and come. This is not a theory class, please understand. Ivat naan ಹೇಳ್ತೀನಿ, no problem, but next class I want you to read and come so that ನನಗೆ ಗೊತ್ತಾಗಬೇಕು ನಿಮಗೆ ಎಷ್ಟು ಅರ್ಥ ಆಗಿದೆ, ಎಷ್ಟು ಅರ್ಥ ಆಗಿಲ್ಲ, where I have to teach and where I have to not to teach. Please read and come, or else this will not work. Nim syllabus ಆಗಲ್ಲ, I'll not be responsible then. Please understand my problem. Fine. So now it is complicated. Pay attention. This enzyme is going in this direction. So each strand mal kuda, it will also go in this direction. There is only one enzyme. There is no two different enzyme. ಒಂದೇ enzyme ಎರಡು ಸ್ಟ್ರಾಂಡ್ ಮೇಲೆ ಕೂತ್ಕೊಂಡಿರುತ್ತೆ and it has to go in this direction only. But problem is only one strand gives you 3' to 5' direction so that it can easily go in the 5' to 3' direction. Another strand's polarity is reverse. Another strand is in 5' to 3'. Avaga ಹೊಸ ಏನ್ ಸ್ಟ್ರಾಂಡ್ ಆಗತ್ತಲ್ಲ that becomes discontinuous because what the enzyme will do, no, every time it goes forward it will pull this. ಇದನ್ನ ಹಿಂಗ್ ಮಾಡುತ್ತೆ. It will pull this strand like this, synthesize the short synthesize the short fragment here and leave the strand. ಮತ್ತೆ ಮುಂದೆ ಬಂದ್ಮೇಲೆ again pull the strand like this, synthesize the short strand, leave the strand. So ಹೊಸ strand ಯಾವಾಗ ಆಗತ್ತಲ್ಲ it looks in bits, small fragments ಕಾಣುತ್ತೆ. Simple logic ಹೇಳ್ತೀನಿ. Again use this two strands. Okay, one strand ಮೇಲೆ enzyme is simply moving like this. So one strand ಮೇಲೆ continuous strand ಆಗ್ತಾ ಇದೆ. But another strand polarity is reversed. ಅದಕ್ಕೆ ಈ ಥರ ಹೋಗ್ಬೇಕಂದ್ರೆ it will simply pull, ಇದನ್ನ ಹಿಂಗೆ pull ಮಾಡಿ ಇಲ್ಲಿ ಸ್ಟ್ರಾಂಡ್ ಮಾಡಿ ಮತ್ತೆ ಬಿಟ್ಬಿಡುತ್ತೆ. ಅಂದರೆ pay attention, I will pull it like this, put a DNTP here and then again leave it. Pull it like this, put DNTP here, leave it like this. Again pull, put here and leave it like this. So ಇದನ್ನ pull ಮಾಡ್ಕೋತ ಹಿಂಗೆ bend ಮಾಡ್ಕೋತ ಹೋಗುತ್ತೆ. And that is why it generates the fragments. And so ಅಲ್ಲಿ gap ಉಳಿದ್ ಬಿಡುತ್ತೆ, which you call it as Okazaki fragments. What is Okazaki? Meaning of Okazaki? What is the meaning of Okazaki? Rihan, ಎಲ್ಲಿದೆಯೋ ನೀನು? ಇವತ್ತು ಯಾಕೆ ಮಾತಾಡ್ತಾ ಇಲ್ಲ? Yes, somebody said. What it is? It is the scientist's name. Okazaki is a scientist name, Japanese scientist I guess, who found out that replication happens in this way. So avun ಹೆಸರು ಮೇಲೆ ಆ fragments ಹೆಸರು ಬಿದ್ದಿದೆ. Okazaki is actually name of a scientist. Okay, so now you know the reason of continuous and discontinuous strand. Is this really clear? In ಮುಂದೆ ನಾನು ಕೇಳಲ್ಲ, okay? I will not ask you whether it is understood or not understood because this is your second chance. Once you have already done your college, ಅಲ್ಲಿ ನೀವು ಡೌಟ್ಸ್ ಕೇಳಿಲ್ಲ, ಇವಾಗೂ ಕೇಳಾಗಿಲ್ಲ ಅಂದ್ರೆ it is your problem then. So I want you to be clear on these concepts. Anybody has any doubts right now? Tell me. Okay, fine. Now who will join these fragments? ಈಗ ಇದು DNA ligase enzyme, yes. So finally you also need DNA ligase that will fill up these gaps. And then you have a one particular full strand. Okay? So this now Okazaki strand is filled up and forms a new strand. ಇದು ಎಷ್ಟು ಎಲ್ಲಿವರೆಗೂ ಕಂಟಿನ್ಯೂ ಆಗುತ್ತೆ? This will go on continuing. ಇದು ಮುಂದೆ ಹೋದಂಗೆ ಏನಾಗುತ್ತೆ? The next DNA would be gone opening. And this will continue until it will complete and reaches the point where it had started. Until then, this process keeps on going. Getting? So, in E. coli, generally it happens in 18 minutes. What is the division or amount of time taken by E. coli to divide? 20 minutes. Adralli, 18 minutes it does only the synthesis or replication concept. If you talk about humans or eukaryotic cells, especially human cell take 24 hours to divide. At 24 hours only, 23 to 22 hours it will do only S phase. Or if you remember interphase, yeah, do you remember interphase? PUII cell division, chapter 10, where interphase was divided into S phase, G1 phase, S phase, G2 phase, then M phase where you have prophase, metaphase, anaphase and telophase. So interphase alli all this katha is happening, and that is why we say interphase is the longest phase. Second ಏನಾಗುತ್ತೆ in the prophase, metaphase, anaphase and telophase, simply in last 1 hour it will quickly divide into 4 phases and forms 2 new cells. So you can now understand replication is such a big, huge, error-free. It should be error-free. Error-free andre? Accurate. So who is going to be accurate? This DNA polymerase should be very, very accurate. Andre, by mistake, suppose it was A here, it should put T. And if it puts C, then this is a mistake. This is an error. ಹಿಂಗಾಗಬಾರ್ದು ಆವಾಗ ಪಾಲಿಮರೇಸ್ halts. Polymerase will hold on, go and check what is the correction. Correction ಇದು ತಪ್ಪಾಗಿದ್ರೆ again it will put the right nucleotide and then move ahead. ಹಿಂಗೋಗಲ್ಲ, ಹಿಂಗೋಗತ್ತೆ actually. Getting? Understanding? Okay, and what is the speed? What is the accuracy of, what is the speed of polymerase? How many nucleotide it puts? ಹೇಳಿದೆ ಆಗಲೇ. Per second. So I think it is beyond imaginable. Lightning speed. Correct? Per second it is putting 2000. And our 2000 base pairs should be error free, okay? What is the human DNA length? 3.2. Double. Diploid idre 6.6. Haploid idre 3.2 into 10 raised to power 9. The 10 raised to power 9 andre, do the maths, okay? I'm not good in maths. How many zeros and how many base pairs should be there? And ashtella madake, if it is taking 23 hours, then it is correct. Yes, it needs so much of time to do all this story. Okay, that finishes your application. Any doubts here? ಮಾಡ್ರಿ ಇದನ್ನ ನೋಡೋಣ. Please raise the cards. I want to see how well you have understood. Raise the cue cards. Let me see. Be careful, okay? If you make mistake here, no problem. Tappidre tappali, no problem. Okay, cards, please. Rihan, are you there? Satish sir, is he there in the class? Yes. Mat maatadilla yake? Ishtella process helade, one answer illa nin kadande. Not understood this chapter, Rihan? Yes, yes. Yake? You find difficulty? Okay, at least this is clear now. Okay, others, Nipani, Raibak, Joseph.
Option B.
Nancy, understood. Option B. C. In which of the following statement the correct regarding the process in E. coli? What is correct they are asking? What? C? Fourth one. The DNA dependent DNA polymerase catalyzes the polymerization in 3 to 5 direction. Okay. Others ಏನು ತಪ್ಪಿದೆ? 3 to 5 ಇದೆ ಇದು. DNA dependent RNA polymerase ಅಲ್ಲ. Okay. ಇದರಲ್ಲಿ ಏನು ತಪ್ಪಿದೆ? DNA dependent DNA polymerase catalyzes in 5 to 3 as well as 3 to 5. Not in both direction. C option check. C option says that DNA polymerase can catalyze in both the direction, 5 prime to 3 prime and 3 prime to 5 prime. True or false? Yes. Be careful, people. This is a recent year question. ನಾನು ಎಲ್ಲನೂ PYQ ತಗೊಂಡಿಲ್ಲ, ಕೆಲವೊಂದು ತಗೊಂಡಿದ್ದೀನಿ. There are some, you have got study material. Everybody got the study material? Okay. Nipani, did you get? Nipani colleges, have you got the study material from myPAL? Teachers, at least you answer me.
Yes, ma'am.
Okay. Raibag New PU, have you got the study material? I wanted to solve questions. That's why I'm asking you. Have you got the study? Do you have? Sikkide nimge? Answer madro. Raibag Keli? Okay, fine. Nan gottilla yavag sigbodu adu, but meanwhile, at least you can try to solve. You can use your phones and check neat questions on molecular base of inheritance hagbittu. Try to solve practice questions, madi. Do the practice. Okay, what am I answer getting here? Come on, answer. A. Option A, option C, option A, option C. Okay. In RNA world, RNA is considered the first genetic material evolved to carry out essential life processes. RNA acts as a genetic material and also as a catalyst for some important biochemical reactions. Being reactive, RNA is unstable. True? These statements are directly there in your NCRT. Check. RNA world ali check madi. The DNA evolved from RNA and is a more stable genetic material. Its double helical strand being complementary resists the changes. True. Where is such option then? 3. 3. Both statements are correct. Correct? Any doubt here? You want me to say anything? Any solving? Any confusion? Raibak people, you are not responding at all, okay? ಹಿಂಗಾದ್ರೆ ಯು ವಿಲ್ ಬಿ ಇನ್ ಲಾಸ್. ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳಿ, ಎಷ್ಟು ಬಾಯ್ಬಿಟ್ಟು ಹೇಳ್ತಾ ಇದೀನಿ. ಪ್ಲೀಸ್, ಇಫ್ ಯು ನಾಟ್ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡಿಂಗ್, ಆಸ್ಕ್ 100 ಟೈಮ್ಸ್ ಐಮ್ ಟೆಲಿಂಗ್ ಯು. ಓಕೆ ಫೈನ್. ದೆರ್ ಆರ್ ಮೆನಿ ಕ್ವಶನ್ಸ್ ಆ್ಯಕ್ಚುಲಿ ಟುಡೇ, ಬಟ್ ಅಷ್ಟು ಟೈಮ್ ಇಲ್ಲ ನಮ್ಮ ಹತ್ರ. ನಾಳೆ ಮಾಡಿಸ್ತೀನಿ. ಡೂ ದಿಸ್. ಒಂದೇ ಮಾಡ್ತೀನಿ. ಡೂ ದಿಸ್. DNA replication semi-conservative was first shown in. Fine. Okay, I'm going to stop here. Rest of the questions I'll continue. Okay, read and come. Transcription, naale transcription. Again, a big concept. Transcription, post-transcription. Very beautiful concept, people. It hengide gotta ee chapter. Once you listen very carefully with a clean and calm mind, you will never forget the process. Trust me. So listen with a very focus. This you will remember for forever. Okay, so let's come back tomorrow or whenever I have a class and start with questions. First, we'll solve the questions on replication and start to the transcription. Okay, give the feedback for Tafat and have a good day, people.